A very good morning to everyone. Welcome back to for a webinar on novel therapeutic approaches for antimicrobial resistance for the second day. Okay. First of all, I would like to all the participants, those who are joined in this webinar, I welcome you all. And also I appreciate your interest to attend this webinar. Thank you for that. So in the Google Meet, only 11 participants are there, so we can wait for a few minutes. Okay, we can start our session. On yesterday, we have studied about that antimicrobial resistance and how do antibiotics works, and also the types of antibiotic resistance and what are the differences between the antibiotics and antimicrobial resistance, and also about the mechanism of antibiotic resistance. So these are the content we have seen on yesterday. In today's session, we are going to see about the factors contributing to antibiotic resistance. So what are the factors which are affecting the antibiotic resistance are? There are mainly 4 factors are there. The first one is the environmental factors, drug-related factors, patient-related factors, and physician-related factors. So these 4 factors which affect to the antibiotic resistance. So the first of all, we can see about the environmental factors. So the environmental factors which affects the antibiotic resistance are, that is due to huge population and overcrowding. So in our country also the population is high. So due to this huge population and also overcrowding, it will affect the antibiotic resistance, and also rapid spread, and it increases the traveling. When we are traveling, if a human being is affected by a disease, and when he or she is traveling from one place to another, so that factors also which affect the antibiotic resistance. And next one is the poor sanitation and the increasing community acquired resistance and ineffective infection control program. So these are the environmental factors which affect the antibiotic resistance, and also it widespread the use of antibiotics in animal husbandry and in agriculture, and also in medicated cleansing products. So these are the factors which are contributing to the antibiotic resistance. These are the environmental factors. And next one is the drug-related factors. The drug-related factors, we can see that fake drugs. So by using the fake drugs also, it will affect the antimicrobial resistance and the quality of the drug, and soaring use of antibiotics and over-the-counter availability of antimicrobials and irrational fixed dose combination of antimicrobials. And also these are the drug-related factors. substandard drug causing suboptimal blood concentration. So these are the drug-related factors which affect the antimicrobial resistance. And another thing is patient-related factors. The patient-related factors you can see here under poverty, lack of education, self-medication, poor adherence of dosage regimens, and misconstitution. So these are the patient-related factors which affect the antimicrobial resistance. And the next one, we are going to see about the combination of drug therapy. So how the drug therapy, the combination occurs in the drug therapy. So this is about treating the infections. Sorry, there are one more slide is I have missed. That is the next thing is the physician-related factors. The physician-related factors, inappropriate use of available drugs. So we are not at all using the drugs in a proper manner. So that is inappropriate use of available drugs and increased poly antimicrobial use, and also overusage of the drugs and of the antimicrobial, overuse of antimicrobial, and inadequate dosing and lack of current knowledge and training. Right. So these are the physician-related factors which affect the antimicrobial resistance. Next, we are going to see about the novel strategies to combat the antimicrobial resistance. So here some novel strategies are there. Here the two approaches can be used to take this problem. The contemporary novel approaches include there are some approaches are there, therapies are there. The first one is the combination drug therapy, and the second one bacteriophage therapy, third one fecal microbiota transplantation, and the fourth one is the antimicrobial adjuvants. It is to combat the antimicrobial resistance. So these are the contemporary novel approaches of antimicrobial resistance. Now we can see one by one. The first one is the combination drug therapy. So the combination drug therapy is about treating the infection. It is set of drugs, not within a single. It is a set of drugs rather than individual therapy. That is monotherapy. And this approach is currently being used in many drug regimens where the causative organisms are more prone to develop the resistance, and also resistance about mycobacterial tuberculosis, human immunodeficiency viral, plasmodium parasitic infections to develop the resistance against these pathogens. And also the principle is maintained to minimizing the probability of the primary resistance to the combination regimen by many folds. For example, we can consider that organism as X has the primary resistance, and the primary resistance has the drug A and B. Let us consider as 10 power minus 6 and 10 power minus 7 respectively. So the drug AB considered as 10 power minus 6, and the drug B is considered as 10 power minus 7. And it is provided that both the drugs lack cross resistance and does not interfere with each other metabolism and pharmacokinetic properties. So the new possibility of the primary resistance of the X organism, X drug combined with A and B, and it is A and B is 10 power minus 6 into 10 power minus 7. So totally we get 10 power minus 30. So this is the combination of the drug therapy. And the principle to rationalize its use in minimizing the probability of the primary resistance. The combination remains by many folds. And there are the combination drug remains usually act by one of the three ways. There are three ways are there. First one is the combination drugs acting on different targets in different pathways. And the second one is combination drugs acting on different targets in same pathway. And the third one is the combination drugs acting on single target but in different dimensions. So it acts as a different target in different pathway, different target in same pathway, and also in single target but in different dimensions. This drug acting on single target but in different dimensions. So these are the things of combination drug therapy. And the next one you can see one by one elaborated. This combination drugs act on different targets in different pathways. Here the modality used against Mycobacterium tuberculosis infection, currently prevalent in many developing nations like India. First 4 line drugs are used in this regimen. The 4 drugs are rifampicin, isoniazid, ethambutol and pyrosinamide. So these are the first 4 line drugs, and these targets are the rifampicin is that RNA polymerase inhibitor, and the isoniazid is enol reductase subunit of the fatty acid synthesis, and the ethambutol is acts as an inhibitor of arabinosyl transferase, which is involved in the cell wall biosynthesis. And the last one, this pyrosinamide, is the main mechanism of the action is poorly understood. And assuming that the bacterium got a chance to develop resistance by changing one of the target, this combination regimen should be effective against at least other 2 pathways, minimizing the chances of the bacterial propagation. The efficiency of the combination regimens is well appreciated by policy makers and also incorporated for the treatment of various infectious diseases. And similar is the case with the various other combinations. The therapeutic strategies such as bacteria like Mycobacterium leprae infection, HIV, Plasmodium falciparum, that is malaria treatment strategies. So these efficacy of the combination will guidelines for the various treatment act as the treatment of the various infectious diseases. Then the next one is the combination drug acting on the different targets in same pathway. So here the drugs which act as the same pathways are the beta-lactam antibiotic, that is amoxicillin, with beta-lactamase enzyme inhibitor. So that enzyme inhibitor is clavulanic acid. It acts as an enzyme inhibitor with the combination of beta-lactam antibiotic. And here the many or gram-positive bacteria produces the beta-lactamase enzyme, and which opens up the beta-lactam, make it ineffective. So it is this bacteria which make this enzyme as an ineffective form. And adding beta-lactamase enzyme inhibitor to the clavulanic acid, and it degrades the enzyme, permitting the drug to act on these organisms. So other inhibitors like the sulbactam and the tazobactam drugs. So this is a combination drug acting on the different targets in the same pathway. And the next one is the combination drugs acting on single target but in different dimensions. For example, here is streptogrammin. It's a class of newer generation antibiotics. It's a new antibiotic. It's a mixture of 2 active molecules, not in a one molecule. It is a mixture of 2 active molecules. These 2 molecules are one non-ribosomal peptide and the other is the polypeptide non-ribosomal. So here one molecule is from non-ribosomal peptide and the other is from the polypeptide non-ribosomal peptide hybrid, and it will bind an adjacent sites in 50S subunit near the peptide transferase center, and they are about 10 to 100 fold. So the foldings of that peptide is 10 to 100 fold more potent as the combination than either molecule if it is used alone or in single agent. So this is about the single target, drugs acting on single target but in different dimensions. The next one is the bacteriophage therapy. The bacteriophage therapy, this phage therapy were being used in therapeutically to treat the bacterial infection, to control the bacterial infection that do not respond to the conventional antibiotics. So it will be used to treat the the bacterial infection. Then it is one of the viable alternatives to the antibiotic. So it is one of the alternative of the antibiotic. And it is important benefit of the phage therapy is that bacteriophages can be much more specific than more common drugs and thus harmless to not only the host organism and to the ecology. Here this bacteriophage is a virus, you all know it. The bacteriophage is a virus that infects and replicates within the bacteria. So these are composed of proteins that encapsulated with a DNA molecule or RNA genome molecule and replicate within the bacterium following the injection of their genome into the cytoplasm. And this property can be used to kill the bacteriophage occupied bacterial cells, forming the principle of this therapy. This is the main principle of this therapy. So originally this bacteriophage therapy was developed by a scientist named as Frederick Twart and Felix de Herle in the year 1915 and 1970. The phage therapy was immediately recognized as an important tool for treating the bacterial infections. And also in British bacteriologist Ernest Hakim reported that the antibacterial activity against the bacterium Vibrio cholerae, which has been observed in the Ganges and Jamuna rivers in India. In India only they observed. He suggested that the unidentified substance was responsible for this phenomena and for limiting the spread of the cholera disease. This unidentified substance is now recognized as the bacteriophage. These bacteriophages have the high responsibility, that is, this bacteriophage have high specificity in killing particular bacteria and leaving the other useful bacteria unharmed. So this property is especially useful while killing the pathogens without alerting the gut flora. So this is the main principle behind it, and also the discovery of this bacteriophage therapy. And the antibiotics being non-specific in their actions, that will destroy common cells in the gut region and commonly seen within the drug fluoroquinolone and leading to the super infection with the Clostridium difficile organism. There are several advantages seen with the bacteriophage therapy over the antibiotic, and small doses of bacteriophages are required. No need to require large doses, high amount of doses. It's enough for small doses required to treat the bacterial infections as they self-replicate in vivo. And this also provides an additional advantage of being less immunogenic as less dose for the foreign substance which enter in the body, which are administered in the body. And the main concept of this bacteriophage therapy is as seen with the antimicrobial host, no place in bacteriophage therapy as single bacteriophage is sufficient to kill single bacterium, not more than one bacterium. It will kill the single bacterium. As phage also continue to participate in the evolution, they keep on adapting themselves and the mutational changes will occur in bacteria and leaving the chances of development of resistant bacteria. So it is also a high therapeutic one. So being specific to bacterial species, development of cross resistance is infrequent. So this is about the bacteriophage therapy. The next one is the fecal microbiota transplantation. So here it is also known as fecal bacteriotherapy or stool transplantation. And this FMT, this process involves the transplantation of fetus from a healthy donor to a recipient. So from the healthy person to healthy donor to a poor recipient or a healthy donor to the healthy recipient also. So this is the process involves the transplantation of fetus from the healthy donor to the recipient. And the active principle behind this therapy is the total resorption of the gut commences by infusion of the source by a healthy donor. By using these various methods, including enema, nasogastric, nasoduodenal and colonoscopic routes. Through this routes, we can exchange this fecal microbiota transplantation. And here the major act of the organism is Clostridium difficile. This Clostridium difficile infection is the leading cause of the antibiotic and health care related diarrhea, and it causes the diarrhea. And fresh infection, if some person gets a fresh infection, that the free stool from the donor is processed in the laboratory and they will dilute it, and it is then sprayed into the large intestine through colonoscopy. So this is the procedure how the fecal microbiota transplantation may occur. The fresh infection or the free stool from the donor is processed in the laboratory and it was diluted, and then it is sprayed into the large intestine through the instrument colonoscopy. And for the small intestine, the liquid is introduced through the nose by using the nasoduodenal tube. So this procedure will take around 2 hours. And this first description of this fecal microbiota transplantation was published in 1958 by a scientist called Ben Eisman and his colleagues. And they treated 4 cases of critically ill patients with fulminant, the disease of fulminant pseudomyomelus colitis, using the fecal enzymes. The antibiotic targets being non-specific kills the both pathogen as well as the gut flora. So even this antibiotic kills both the pathogen and also as well as the gut flora. And this antimicrobial action on gut flora kills the organism, leaving behind few resistant strains of Clostridium difficile bacteria. And the drugs, the antibiotic which is most commonly used in this condition is fluoroquinolones, are the most common antibiotics leading to this condition, and the clindamycin and beta-lactam antibiotics are also the contributing agents. And these now this Clostridium difficile bacteria strains are grow unchecked and which are referred to commonly used the antimicrobials leading to the pseudomyomelus colitis. And this therapy was transplants from the gut flora, that is from healthy donors feces, and restores it in the recipient colon, and displaying the selective growth of the virulent strains of the Clostridium difficile bacteria, and decreasing the number, ultimately breaking the cycle of the recurrent disease, the Clostridium difficile bacterial infection. And this minimizes the repeated antibiotic use, which in turn reduces the risk of the antibiotic associated resistance. So here this common antibiotics used are the fluoroquinolones and the clindamycin and beta-lactams are the contributing agent, actors and agents. And the current guidelines here from the infectious disease from America, the vancomycin and metronidazole base remains, but the therapeutic effects are limited by high recurrence rates because of the development of the resistance. Then the next one is the antibiotic adjuvant. The antibiotic adjuvant are composed of that do not sees the fair remissions of the deficiency in the existing antimicrobials for the resistance of development and also the complementary bactericidal mechanisms, and it inhibits the signaling and the regulatory pathways or boosting the host response to the bacterial infection. By using this, the discovery of this new and effective antimicrobials is the ideal approach how to combat the issues of antimicrobial resistance. And also it seems a fair solution for the deficiency in the existing antimicrobials for resistance development. But the development and the marketing approval of these drugs and the US, the Food and Drug Administration, they have not matched the pace of development of antimicrobial resistance. So the best practical strategy is to modify the existing drugs to make them more useful and potentiate their effectiveness. And it will increase the effectiveness of the drug, and this process is more economical. By using this microbial adjuvants, the emergence such approaches that aims to improve the efficacy of the existing antibiotics and for suppressing the emergence of resistant strains. So these are the compounds that make the bacteria is more and more susceptible to the antibiotics. So these compounds typically don't possess any intrinsic antimicrobial activity, but those with such an activity can also be considered as an adjuvant. The generic mechanism of this actions are here. Some mechanism of actions are there. First one is inhibition of an antimicrobial resistance elements, enhancing the uptake of antimicrobial in target cell and nullifying the effect of efflux pumps, and also by promoting the bacterial oxidative stress. The inhibition of this antimicrobial resistant elements, here the addition of beta-lactamase inhibitor to the beta-lactam antibiotic. So thus preventing the dissociation of the beta-lactam ring. So it will cleavage the beta-lactam ring and maintaining its efficiency against the gram-positive bacteria. Only in the gram-positive bacteria it will take place. Another notable example, that is silicistatin and dehydropeptidase. It acts as an enzyme that degrades the beta-lactam inhibitor. These adjuvants supplements to the action of these compounds. So it will inhibit of the antimicrobial resistance elements. And the next one, the enhancing the uptake of the antimicrobial in target cell. Here the colistin. So it is also known as polymyxin B. It is a polycationic molecule with both lipophilic and hydrophilic ends. And it is an antibiotic whose use itself is limited due to the nephrotoxicity. But in this action, as a membrane detergent can be used as an adjuvant, and the principle is while using as a low dose, and this limits the toxicity and facilitates that the penetration of concurrently administered hydrophilic antibiotics such as rifampicin, glycopeptides and tetracyclines. So this principle can be further exploited to target the lipopolysaccharide layer of the gram-negative bacteria. So it is enhancing the uptake of the antimicrobial in target cell. Then the nullifying the effect of the efflux pumps are the main mechanism behind this, the development of the resistance to the antibiotic, that is tetracycline, in by its expulsion from bacterial cells from efflux pumps, through efflux pumps. This approach to achieve this effect is by administrating structurally the similar compounds are present which competes with the tetracycline for the efflux pumps, and it minimizing the efflux of the tetracycline compound. So hence this bringing down the development of the resistance. So this tetracycline brings about the development of the resistant process. This same mechanism can be used in the quinolones and aminoglycosates drug. And this disruption of this biotin bacteria from sessile group is called biofilms as a protective screen to the antimicrobials. And this factor was later recognized as a mixture of, that is D-amino acid, and this can be used as a cobalt promoting antimicrobial resistance. In clinical practice, by co-administrating the D amino acids in the broad spectrum of the antibiotics, and it gives that against the sensitivity of the bacteria. So this is the nullifying the effects of the efflux pumps. And the next one is that by promoting the bacterial oxidative stress. This here the telluride, so it is also known as tellurium oxanate tellurate. It is known to cause high levels of oxidative stress in bacterial cells. So this antibiotic will cause the high level of oxidative stress in bacterial cells, and this antibiotics are acting by interfering with the cell wall. That is, the cell wall contains the ampicillin and subtoxamine, or some proteins that is tetracycline, chloramphenicol, gentamicin. These are used in the sublethal concentrations of the telluride, and the killing action is potentially in the folded form. And this possible mechanism, other than the reactive oxygen species, it will generate or damage the metabolic enzymes, and it will metabolic enzymes, that is glutathione depletion or lipid peroxidation will take place. So these are the generic mechanisms, mechanical actions of the adjuvants act as the includes. Then the next one is the antimicrobial peptides. So these antimicrobial peptides have been identified from many proteins, like hydrolysates, especially from males, and the most well studied are the lactoferrins, which are derived from the boy serum and also the human lactoferrin. Additionally, this antimicrobial peptides is a few antibacterial peptides have been identified from the alpha S1 casein and S2 casein, and also it acts against different both gram positive and gram negative, and also yeast and fungi. So these organisms, it acts against the microorganisms. The antimicrobial peptides act against the microorganisms. The main mechanism of this is the destruction of the normal membrane permeability is at least partly responsible for the antimicrobial mechanism of the lactoferrins. And this antimicrobial peptides is also known as host defense peptides, and it is a part of the innate immune response found among many organisms, and they present significant antibacterial, antifungal, antiparasitic, and also the antiviral activity. And usually these molecules are composed of 10 to 50 amino acid residues, and it is arranged in different groups depending on the amino acid composition, based upon their amino acid composition, size, and their conformation. And these protein molecules have a weak intrinsic antimicrobial activity, but it potent and broad immune modulatory activity when the host cells are invaded by bacteria or viruses, and this demonstrates their potential as a novel antimicrobial agents. And also they have very specific properties, including their amphipathic secondary structure. Due to their small size and positively charged, they bind rapidly to the biological membrane of the organism. So these are the very fast killing microbes, and a property that does not allow the development of the resistance against these particular compounds. And this represents a class with a broad spectrum of about the activity against this bacteria, both gram positive and gram negative. And also these antimicrobial peptides are also produced by all known species, including peptides from bacteria and peptides from fungi, from hydra, insects. For insects, mastroparan and moricin, these are the antimicrobial peptides which appear from the insects. And mammals, the mode of action of antimicrobial peptides is not very clear, but they mainly act by targeting either cell membrane or their cell wall or the intracytoplasmic molecules. So additionally, they also act by neutralizing the lipopolysaccharides and prevent the immune-mediated response for the further development. And this antimicrobial peptides act by multiple modalities. These include that lipopolysaccharide neutralization or disaggregation, induction of membrane permeability, inhibition of cytoplasmic proteins related to cell division or survival, and inhibition of macromolecular synthesis through interaction with nucleic acids, antibiofilm activity of the antimicrobial peptides against the biofilm and multi-drug resistance bacteria. And these acts currently available in the conventional antibiotic by enhancing the permeability of antimicrobials by their membranolytic effect, reducing the dose and reducing the toxicity of the drug. And this strategy is presently in infancy stages, and more studies are needed to validate them for their usage in clinical practice. These can be newer generation for the antimicrobials for combating the multi-drug resistant microbes and preventing the biofilm production. And the next one is the classification of antimicrobial peptides. The classification, these antimicrobial peptides are classified into 4 different peptide structures, which are alpha helical, beta sheet, loop and extended form. In the alpha helical, that manganine antimicrobial peptide contain this manganine compound, and in the beta sheet contain protegrine 1, and the loop containing, that loop is otherwise known as mixed form. It contains the protegrine 1 compound and the extended containing indolizidine, and the beta sheet contains the defensin and the human. So these are in different structure. And the next one is the properties of the antimicrobial peptides. The properties within the antimicrobial peptides are the antibacterial, anti-tumor, and antiviral, angiogenic, and it also heals the wound, anti-inflammatory activity, and immunomodulatory activity. So these are the activities under the properties of the antimicrobial peptides. And the next one is the antimicrobial susceptibility test. And this antimicrobial susceptibility test, it is very important for treating the infectious disease and monitoring antimicrobial resistance in various pathogens. And to accumulate this epidemiological information on the resistance of the microorganisms for public health, and it is a determination of at least the amount of an antimicrobial chemotherapeutic agents. It acts as a chemotherapeutic agent that will inhibit the growth of the microorganisms in vitro. And some methods are there, the antimicrobial susceptibility test. In this method, there are 2 methods are there: disk diffusion method, MIC method, and one more method is steam method. And in this disk diffusion method, we are using, there are 2 types: Kirby-Bohr method and Stokes method. And the minimal inhibitory concentration method, we are using that. In this method, it is classified into 2 types, that is broth dilution method and alpha dilution method. So these methods are using the used to for the antimicrobial. These tests are used for the antimicrobial susceptibility. So by conclusion, this antibiotic resistance has increased in worldwide in bacterial pathogens, leading to the treatment failures in humans and animal infectious disease. And this resistance is against the antibiotics by pathogenic bacteria is a major concern in the anti-ineffective therapy of both humans and also the animals. And this bacteria are able to adapt rapidly to new environmental conditions, such as the presence of antimicrobial molecules and the consequent resistance, and they increase with the increasing exposure to antimicrobials. And the serious concerns about this bacterial antibiotic resistance from nosocomial community acquired nosocomial infections that community acquired and also foodborne pathogens have been growing for a number of years and have been raised at both national and the international. So these modalities form the hope for the future to probe the development of the antimicrobial resistance. So this is about the antimicrobial novel therapeutic approaches for antimicrobial resistance. Thank you. If you have any queries, please put in the chat box. Okay, thank you. We will send the feedback link form and you have to fill that form and get your e-certificate. Okay. Yeah, in the chat box there are questions there. Use some examples of antimicrobial adjuvants. Examples of antimicrobial adjuvants are that is beta-lactamase inhibitor, that is clavulanic acid, tazobactam and sulbactam. And also the antibiotic amoxicillin, which has been commercialized. Okay. Okay, we will send the feedback link to your mail ID, and also in the chat box we have send the feedback link. You have to fill the form and get your e-certificate. And in future we will conduct various events regarding webinar, workshops and virtual programs. So kindly cooperate with us. Okay, again we have sent the feedback link in the chat box. Okay, thank you once again. Thank you for all the participants in this webinar. And also, if you need more updates from our First Bio Research Lab, please subscribe our channel First Bio Research Lab, and for more updates, please connect our WhatsApp group. We will send the WhatsApp group link also. Okay, thank you one and all. Thank you.